Dear audience, my name is Lucien Lemaitre, and I'm here to give a brief summary of my two really different research fields from year 2021 and 22. To start with, I would like to tell you that last December I took part in the Hungarian qualification round of the International Chemistry Tournament. And for this event, I carried out fundamental researches at László Németh Secondary and Grammar School with the supervision of Dr. Eszter Arany. The perspective of this work was the investigation of the possible development of a fail-safe cleaning agents for households, where every major chemical is spiked with a substance that creates a warning signal in case of a harmful interference. The direction of the research was based on the recognition that all the harmful substance formation was dedicated to strong oxidizers like bleaching agents, for example, hypochlorite or even peroxide. So therefore, I started to examine several types of redox indicators that could possibly be added to the prototype systems in order to produce a color change reaction and thus a warning signal. So lastly, I came up with two different types of indicators, DPA or diphenylamine and LMB like leukomethylene blue. And as both creates a definite blue color change reaction via oxidation, they found to be proper substances in the prototype system. And this elaborated prototype system and the explored difficulties so far provide us the opportunity for further developments. And now, moving to topic number 2. Since 2019, I've been investigating the field of Cardination Chemistry in the Bioorganic Research Group of Szeged, led by Dr. Tamás Gajda at the Department of Inorganic and Analytical Chemistry at the University of Szeged. And these organic complex compounds, also called as metalloorganic compounds, are used in clinical practice for a really long time. And without the demand for completeness, I'd like to highlight some. For example, instead of insulin or against ulcer, inflammation, high blood pressure, or even against parasites. And I would like to highlight the last one, even against cancer. And referring to this last one, I'd like to point out two specific anti-cancer agents, the other cisplatin and the newer generation NAMI-A. And it's clear that both contains platinum group metals, as these platinum group metal complexes were identified as potentially anticarcinogenic drugs. Concerning the aspiration of our research group, the long-term perspective is the investigation of the possible development of a trifunctional anti-cancer agent. I mean, we try to prepare a complex system where carcinomas are attacked on three fronts. On the one hand, it has been determined that alpha-V beta-3 integrins have a higher concentration on the surface of these tumor cells that are responsible for the faster angiogenesis of tumor cells than normal cells. So therefore, we start to research a peptide derivative that contains an RGD, an arginyl glycyl aspartic acid sequence that is in charge of binding to this alpha-V beta-3 integrin in order to modify its conformation, thus stop or at least decelerate this fast angiogenesis. On the other hand, the other parts of the peptide beside the RGD sequence could be utilized for targeted drug intake. To tell the truth, any kinds of anti-cancer agents could be possibly attached. However, our research group examined ruthenium and rhodium binding subunits, as these subunits were identified as strongly anticarcinogenic agents when they enter to the tumor cell cytoplasm. Through they attack both polynucleotides and some types of enzymes at the same time. And the possible way to release these rhodium and ruthenium metals from their coordination field is their substitution with free copper-2 ions. And free copper-2 ions are found also in the tumor cell cytoplasm in a higher concentration than in normal cells, and they also play a really essential role in the previously mentioned faster angiogenesis. So the elimination of the free copper ions from the tumor cell cytoplasm has a further antiproliferative effect. However, it's really important to point out that targeted drug intake is mostly really a complicated issue because of the high possibility of interference between our future anticancer agent and the compounds found in blood plasma that are mostly proteins and peptides. So therefore, an alternative research direction has been developed in the last decades because there was a clear demand for the thematic thermodynamic examination of these metal protein and metal peptide systems. And as it's nearly impossible to extract thermodynamic data from the bigger sized protein metal systems, we use, beside the endogenous peptide investigation, model compounds in order to mimic the metal binding sites of proteins that has a bigger size. And it has been already determined that the most significant binding units to these mentioned platinum metals in case of peptides and proteins are histidine subunits through their imidazole ring. And as for the metal, we currently examine the last described rhodium beside the ruthenium, and the extreme slow reaction kinetics of its aqua complex led to the displacement of 3 of its water molecules to an aromatic cap. And this derivative compound, called a half sandwich compound, has still slow but way faster kinetics than the alpha complex has. And to tell the truth, this cap could vary. There are loads of aromatic caps that could be possibly utilized. However, we recently and currently examine CP shortly, or pentametal cyclopentadienyl. And beside the binary investigations, we also carry out some ternary simulations as well in order to mimic the possible interaction between our future anticancer peptide agent and our currently investigated ones. And therefore, we use the strongly coordinating bipyridyl and ethylene diamine as model compounds that we try to mimic with these strongly coordinating ligands the future anti-cancer agent. And during these simulations, we examine whether these strongly coordinating compounds are displaced from the coordination field of the rhodium as the investigated peptide has an even stronger coordination affinity to the rhodium metal. And the results of these simulations give us or provide us and predict us the possible interference, the type of interference between our future anti-cancer peptide agent and the currently investigated ones. And currently, 2 endogenous peptides are examined by us, beta-alanine histidine, or shortly carnosine, and glycylhistidylysine, or GHK. Both of these compounds were described in presence of rhodium CP and copper-2 ions. And the significance of our results is based on that both of these compounds can be found in the human body in a really high concentration. Thus, the interference between them and our anti-cancer agent is for sure. And they also takes part in some important metabolic processes, like both of them has the pH buffering, an antioxidant and an anti-inflammatory effect as well. And on top of that, carnosine found in some researches that have potentially anti-carcinogenic effect, mostly related to mug cancer. And GHK has a tissue regenerating effect, and it also takes part in some epigenetic modulating pathways. But lastly, it's really important to emphasize that both compounds are really important metal chelators in the human body as well, and especially GHK that can be found in blood plasma in vast majority in its 1:1 copper-2 complex form. That structure is represented here on my figure. And lastly, I'd like to say thank you for all my supporters, to my two great supervisors, and all the supporting institutes as well. I'd also like to thank you for your great attention.
Thank you very much for your brilliant presentation, and I also would like to ask Csaláné Böngyikedit to come up to the stage, and we would like to give the certificates for both of you, and because of course it's always need students and mentors together, so congratulations for both of you.